In the shadowy underbrush of European forests, the 𝗕𝗥𝗢𝗧𝗘𝗥 stealthy, parasitic arthropod has long been an unwelcome predator. Yet within its salivary glands lies a biological treasure that could transform how we treat one of the most devastating cerebrovascular conditions, hemorrhagic stroke. In a bold act of scientific curiosity and translational innovation, Belgian biotech startup 𝗕𝗥𝗢𝗧𝗘𝗥 has unearthed a powerful secret from this tiny arachnid. Their lead candidate, 𝗕𝗥𝗏𝗏101, is a first-in-class biologic inspired by a tick's salivary protein that manipulates host homeostasis. This molecule originally evolved to prevent clotting and immune detection during a 𝗕𝗥𝗢𝗧𝗘𝗥 blood meal. It's now being repurposed as a dual-action neurovascular therapeutic to treat thrombosis and neuroinflammation in patients with hemorrhagic stroke. The science, taming hemostasis without bleeding risk. Unlike ischemic stroke, where clots obstruct cerebral vessels, hemorrhagic stroke involves the rupture of weakened blood vessels, often leading to life-threatening intracerebral bleeding, brain swelling, and secondary neuroinflammation. Current treatments are limited, with a narrow therapeutic window and significant bleeding risk. 𝗕𝗥𝗏𝗏101 targets kallikrein-kinin system KKS pathways and neutrophil-driven inflammation, reducing both clotting and neurotoxicity, but crucially, without impeding primary hemostasis. The active moiety, a recombinant protein derived from the 𝗬𝗥𝗡𝗧𝗔𝗧𝗏 exhibits anti-thromboinflammatory effects by selectively modulating the contact activation pathway, allowing natural coagulation processes to proceed normally in damaged tissues. This makes 𝗕𝗥𝗏𝗏101 one of the few drug candidates to mitigate inflammation and clotting without increasing hemorrhagic risk. A paradigm shift in neurovascular medicine. Milestones and momentum. The early science was validated in robust preclinical studies where 𝗕𝗥𝗏𝗏101 significantly reduced infarct volume, brain edema, and inflammatory cytokine release in rodent models of stroke. Following this, 𝗕𝗥𝗏𝗧𝗘𝗥 completed a phase 1 study demonstrating favorable safety and pharmacodynamics in healthy volunteers. In June 2024, the company launched a randomized, placebo-controlled phase 2 trial in patients with acute hemorrhagic stroke to test efficacy in real-world clinical settings. To support this effort, 𝗕𝗥𝗏𝗧𝗘𝗥 secured €12 million, $13 million in series of funding in November 2023, led by European venture capital groups. These funds are being used for clinical trial execution, GMP manufacturing, and biomarker strategy development. Due diligence narrative. From an investor or strategic partner perspective, 𝗕𝗥𝗏𝗏101 stands out on several fronts. The scientific novelty is high, being the first therapy to leverage a 𝗕𝗥-derived protein for stroke. The target biology is strongly validated, focusing on the kallikrein-kinin system, a well-documented contributor to neuroinflammation and thrombosis. The unmet medical need is critical. Current stroke therapies inadequately address the dual burden of clotting and inflammation, especially in hemorrhagic stroke. The intellectual property IP position is robust, with granted patents in the US, Europe, and Asia covering composition, sequence, and therapeutic applications. On the clinical side, 𝗕𝗥𝗏𝗧𝗘𝗥 is advancing with discipline, having completed a phase 1 study and progressing into phase 2 with a clear biomarker plan. The management team brings deep translational experience, including ex-Johnson & Johnson executives and academic leaders in neurology and protein engineering. Overall, the exit potential is moderate to high, especially for CNS-focused pharma players seeking pipeline expansion in neurovascular or inflammation-related domains. Looking ahead, partnerships, biomarkers, and platform expansion. Beyond hemorrhagic stroke, 𝗕𝗥𝗏𝗏𝗧𝗘𝗥 is evaluating 𝗕𝗥𝗏𝗏101 for other indications including traumatic brain injury, vasculitis, and even long COVID-associated microvascular inflammation. The company is exploring preclinical partnerships around companion diagnostics and biomarkers for patient stratification, intranasal or pre-hospital formulations for faster delivery, and expanded pipeline opportunities for other thromboinflammatory diseases. Final word. From predator to protector, The tick's venomous trickery is being turned against one of humanity's most lethal neurological threats. Bioxid's 𝗕𝗏𝗏𝗏 is not just a drug. It's a story of nature's complexity decoded for healing. If successful, this innovation could mark a new frontier in biologics, one where the silent weapons of parasites are re-engineered to save lives.